Yes, hello, good afternoon. Series on our educational Omega Bio-Edutainment. This afternoon, a kind of review into all my past videos, which we already have at this platform. Already, we have a lot of let me say we have more than almost 10 videos on micro videos on microbial genetics. So, looking at far the behind the camera scene of this revision at first, just that it's just going to be like, it's just going to be like, although the way it is appearing, it is actually appearing as if it's just unorganized anyway. But the just to allow the just to allow viewers to make to have a, viewers to have a glimpse into what we have done so far in the microbial genetics. Okay, anywhere in the whole world, whenever microbial genetics is mentioned, these are part of what should the content should be, especially at the elementary stages of this. So elementary stages of this. So today's topic is actually being captioned with gene expression, said we emphasize on transcription, translation, gene regulation, proteomics, genome sequencing, that is molecular of recombinant DNA anyway, so to say. Now, looking at the board, from what I can see, I can actually see gene expression. Don't forget that what do we actually mean if we talk about gene expression? Gene expression is nothing, but I just like I told you, I normally tell you, and I said in the uh, online video that online video that one of the reasons why gene must continually being expressed is that some genes are more important than others. It's just like the theory of use and disuse, whereby any gene that is a functional gene that microorganisms tend to use more often, so that gene must continually more often. So that gene must continually be expressed from the ones that are expressed from the ones that are not that too functional should continually be repressed. So that is why this topic is actually captured gene expression and gene regulation. So it's just gene regulation. So it's just how genetic information from double-stranded DNA is getting transcribed into single-stranded ribosomal RNA, and later on this will be will be translated in form of a protein synthesis into functional polypeptide. Don't forget that if we mention polypeptide is still the same thing as we are looking at protein. And from the earlier video, the last part of the last, which is a post-translational effect, is a cleavage immediately after adenylation, immediately after phosphorylation and glycosylation. So the last step under that is talking about cleavage. It is until when those one are cleaved before the uh, gene will become functional. That is a functional polypeptide. So, and that is exactly what we have. So, having said this, you see, just like I have always been saying that gene, just like this, what gene expression is DNA to RNA, then to functional polypeptide. And I said that if there is no cleavage, if there is no cleavage of a phosphorylated polypeptide, if there is no cleavage, that means this protein cannot become functional. Okay, then in the process of a gene expression, we made mention of the stages include initiation, elongation, termination, and a kind of a transcriptional modification. This modification involve the first one is capping. So, and capping is nothing but adenylation, that is addition of multiple adenine at the 5' end is called adenylation. So after that, there will be splicing, especially if splicing will only take place if we are talking about eukaryotic organism. So don't forget, introns are non-coding regions, non-coding, whereas introns are coding regions. So you have to take note of this. So don't forget the very important, or let me say, call them key enzyme or key components of transcription. The transcriptional factor and the RNA polymerase, they will bind. They are going to bind at the promoter initiates the process of transcription. And by the time this one binds, you know it's going to bind. So you know what is going to take place is that the process of transcription will continue. It's going to continue until we now have unwinding of the double helix structure of DNA. And don't forget that at that point in time, even at elongation level, we have uracil replacing where we have thymine, because this time around we are actually looking at the production of a messenger RNA, which is quite different from the DNA counterpart. Then again, also here we have okay. So now under gene regulation, there's something we call operon. And what do we mean by operon? Operon is a region of the genome. Is a region of the genome of microorganisms that code for multiple genes. That code for multiple genes, and these multiple genes are regulated together. That is under the control of one operon. An operon can actually direct, control, or regulate multiple genes, not only the genes whereby you have so it's like that is the advanced way of controlling the gene. For example, now it's just like when you have a switch. If you have a switch box that is controlling maybe many electric bulb, like 10, 15, and even more than that. So the switch box is going to operon, and that is what must be switched on. It must be switched on before there will be initiation of a transcription. So it's just going to be like a straight line, whereby on that straight line we have this is the operon. This is the operon. So on that place we have a promoter, we have enhancers, and so on and so forth. That is operon. And whatever we call, if we now say lack operon, that is very possible, very common in E. coli. What is it doing? It's lack operon regulates the breakdown of lactose in the system. So if we now have lactose in the system, the lack operon is the one that is going to control it, so that the lactose will not get accumulated, and there's going to be any problem at the end of the day. Although they need lactose to carry out many and to carry out many metabolic processes, especially do with glycolysis and cellular respiration. Then you will also need to familiarize yourself again with what is called plasmid. Plasmid is just the extra chromosomal material that is actually found in some microorganisms, and the plasmid perform special function like antibiotic resistance. Then other they also have other survivability tendency. Then we also have mitochondrial RNA, or believed that the DNA, sorry, the mitochondria is such also an independent, semi-autonomous entity or organelle that has its own genetic material. So there are some genes, mitochondria, and there are some that are located on the plasmid, whereas some are, the other one are located in the main chromosome. That is the main DNA of the chromosomal, which is chromosomal DNA. And mutation is nothing but what? A sudden change in genetic makeup. It could be frame shift. It is a frame shift is whenever we now have I think we are going to come across what a frame shift mutation is. So anyone that is as a result of insertion or addition that leads to shifting of the messenger RNA, or that leads to the shifting. That. So then we also have codon. Codon are set of nucleotides. These are set of nucleotides that code for example, AAA is actually coding for something. And when we talk of horizontal gene transfer, that is a kind of genetic exchange, between A and B, that is of the same species. That is why they call it horizontal. And you know it is like this, it's horizontal. If it is vertical, that is from the parent to the offspring. Okay. Then conjugation. Conjugation is like the sexual form of reproduction that is found in some bacteria. It's not all bacteria that can undergo what is called conjugation. So then transformation, from the word transform, that is where a kind of dead DNA become reactivated again. So as a result of that, and transduction often involve the activities of phage, that is viruses, that is transduction. And replication means that just from the word duplication. So before any cell will undergo cell division, that cell must the DNA must be replicated or duplicated for it to have. And this is another point which is called metabolomics. We are now in the world of omics. Metabolomics is the application of bioinformatics to be able to study some metabolic pathways in this. And we have genome. Genome is the totality of the genotype in an organism, and genetics is the study of heredity and variation. And whenever you talk about redundancy, there are some part of the characteristics of redundancy in the genetic code. Some of the genetic code are said to be redundant. And what do they mean? The protein synthesis, they produce protein in the presence of a mutation, whether there is mutation or not. This one will we careless about whether there is a mutation. So that is called redundancy. And again, Recombinant DNA technology. Recombinant DNA technology, another word for recombinant DNA technology is genetic engineering. That is manipulation of the genetic material, in particular DNA, for their ability to be able to perform a desired function. For example, in the genetically modified plants, GMO, so if you decide to have a breed, a plant breed of maybe like banana now, if you want to have a GMO that is genetically modified, you want to improve that banana by the use of recombinant DNA technology. What you do is you go and get the gene that is actually being responsible, that is going to be the gene of your interest, and putting it into the main DNA, so that by the time the DNA also replicates, that is going to also replicate along the line, and you will go and look for a suitable vehicle, the suitable vehicle or phage that is going to multiply it for you, so that at the end of the day, within a very short period of time, you now have the gene of interest already being multiplied or being multiplied, so that you'll be able to have more than enough. And frame shift mutation, I've actually explained that we have histones. Histones, what do we, histones is just the protein that is wrapping, that is covering the DNA. So DNA does not actually exist naked. So especially in eukaryotic organisms, they exist in company with histones, which is a protein. And that protein plus histone is what is called chromatin altogether. And DNA sequencing is a technique which is which has gained a kind of popularity among researchers today. So they started with Sanger principle, whereby you have to be adding the nucleotide manually, but now we have moved to next generation sequences. A special example of that is Illumina sequencing. So you don't need to actually bother yourself about that at this level. And don't forget translation. Translation also takes place with initiation, elongation, and termination before you now have post modification. So we have RNA. RNA will be produced three different types of RNA. Transfer RNA from the word transfer is involved in transferring. So you can see here is where you have anticodon. Okay, so it's the one that will be transferring the amino acid from ribosome down to the ribosomal RNA to messenger RNA. Okay, so I've explained what genetically modified GMO is. So proteomics, that is the bioinformatics application of protein structure, and also using it to determine their function. And equipment they normally use in studying protein is called mass spectrometry, which is very, very important. Then don't forget what we mean by gene. Gene is a segment of DNA. So, and the genetic code, in the earlier video, there's a video that I actually dedicated to how can you by heart without by heart, so that you'll be able to know it, if not all, at least you'll be able to know how genetic code is being organized, and how you can actually predict and get the exact genetic code that is going to be paid for, you're going to do for. Okay, then looking at this diagram, we can see a kind of transfer from one cell to another one, and this is as a result of the organism that contains pilus. So pilus is known as a kind of used for sexual reproduction in bacteria. So this one will be transferred from this one. So through a process which is called conjugation. And just like I said, anyone that involved, this is a phage, this is a viral, this is the one that is called transduction. So, and transformation is reawakening of the dead DNA. So, so just like I said, you see, this is just a kind of a revision that is going to help us to be able to in the earlier videos, and so that we'll be able to make use of it and attempt the quiz that will be deposited at the right time on the in the comment section of this division 002. So also, just like I said earlier on, let me use the camera to now capture everything that we have. Let me just mention them again. We have seen gene expression. We have seen how DNA will get into RNA, and the RNA eventually will go and give us a functional protein, what is called a polypeptide, after it has been cleaved. Then we also talk about central dogma theory. Central dogma theory is the theory that believes from DNA to RNA then to protein. That is a central dogma theory, and this is very, very important in um, especially in molecular biology. So very, very important. We look at transcription. We see transcription proceed through initiation, elongation, termination, and we also have a transcriptional modification, which include splicing, as well as splicing, which is exon. We have seen exon, we've seen introns. Okay, so then we will look at adenylation, that is capping of capping, that is addition of polyadenylation to the protein that is going to be produced at the end of the day. Okay, now again we now look at operons, which I said is a region of the genome that codes for multiple genes regulated, and which become regulated all together. So I told you this is just very similar to the switch box, the switch box that controls many. So we also mentioned lack operon that is found in E. coli. So this one is the one that regulates the breakdown, breaking down of lactose in the system. Once the lactose is becoming accumulated or necessarily being accumulated, the lac operon will be switched off, and what is lac operon is going to do is going to mop up the concentration or the amount of lactose that will be found in the system, because you know the implication of having too much lactose in the system. So we also made mention of plasmid as extrachromosomal material that is found in some genes, and we have seen that it is one that is actually being used by bacteria for for resistance. Okay, so mitochondria RNA, I said mitochondria is another organelle that is semi-autonomous, that they also carry out their own independent replication or transcription, irrespective of the what is happening in the cytoplasm or the nucleoplasm. Okay, so that is this. Mutation is just that different types of mutation. We have frame shift mutation, we have insertion, we have deletion, and we have genetic recombination, and so on and so forth. Transduction, the one that has to do with okay, I said codon. Codon are set of they are set of triplet bases or nucleotide that will code for one a specific amino acid. So, and horizontal gene transfer, just like the illustration you have between A and B, the same transfer of genetic material among species of organisms. Okay, so that is that. Then we also consider conjugation, that is sexual reproduction in microorganisms. Transformation, just like from the word transform. So the organism is already dead, the DNA, for example, now let's say where you normally dispose laboratory where you normally dispose laboratory waste. So you see by the time you are disposing them, there are some of the DNA that will be dead one way or the other, even if no matter what, but they will still remain around in the environment, but they will be waiting for one opportunity to be able to come back to life. So transformation is just like when a DNA, when a new organism or a new bacterial organism which is still living take up the DNA of the dead DNA, and the DNA now become integrated into the main DNA of the host. So that is what is called transformation. Anyone that has to do with phage, that is viruses, that's where you have transduction and phage when it comes to the issue of phage. So then again, we also look at replication, that is a process whereby DNA duplicates itself. So duplication. So and this one actually take place before growth. So in order for growth to take place, there must be the DNA must first of all to replicate. So that at the end of the day, we are going to have um, four DNA that we are going to have 4 from 2 DNA that is already being produced, and replication will proceed in semi-conservatively. So we are going to, although we have not actually dedicated any video for this replication, so we are going to have a different video that it will be for replication. Then we also look at metabolomics, that is an application of bioinformatics or the word of omics, metabolomics, to be able to study some of the metabolic pathway in microorganisms. And this has also enabled us to be able to or even identify new strains and new species of microorganisms. And we also look at what a genome is, that is the totality of the genetic makeup of an individual. And we know that genetics is a branch of science that studies, and we also look at redundancy in genetic code. That is when protein synthesis proceeds irrespective of whether there is a mutation or not. That is redundancy in that kind of genetic code. So we talk also about RNA recombinant, recombinant DNA technology. Okay, recombinant DNA technology, and which we have seen so far. Yeah, we have seen this so far. Recombinant DNA, that is a manipulation of, is a process whereby we manipulate a certain segment of DNA to get a desired result. So that is that. And again, don't forget the function of transcriptional factors and RNA polymerase, promoter, as well as an enhancer. That is during the process of gene expression. So we also look at this diagram of genetics between two species, genetic exchange between two species as a result of through their pilus. So through the process called conjugation. So we can see from the one that is up, so you can see that the DNA, the arrow is pointing down, wanting to get integrated into the host DNA, the main host DNA, and that is going to carry on the process of transcription. Okay, the process of transcription, and it will continue like that. Then transduction, I've said that as well. So gene regulation. So don't forget that how are genes being regulated? I told you earlier on that if you have functional gene production without getting it regulated, it's like movements on your vehicle movement of a vehicle, or you have a vehicle that is very, very good, and the vehicle unfortunately do not have a brake, so there's nothing to pause, nothing to stop the car, so you continue to go, you move and move indefinitely without non-stop. So that is that. So that is one of the reasons why gene must be regulated. And we also made mention of purposes of DNA sequence, purposes of DNA sequence, whereby we look at different types of, yes, different types of sequences. So whereby we talk about, yes, we talk about purpose of DNA, we talk about Sanger sequencing, next generation sequences, and an example of next generation sequences is Illumina sequencing. So, and translation, which is a process by which the messenger RNA gets translated, gets converted into protein. So that is that. And we are going to RNA. We have a transfer RNA, we have ribosomal RNA, as well as a messenger RNA. Although there are other types of RNA, but these three are the major contributors in protein synthesis. So we also, this course we also lead us to proteomics. That is the structure of protein, the study protein by knowing their structure as well as a function, believing that whenever we are able to know or to use bioinformatics to be able to predict the three-dimensional shape of a different protein, that one can go a long way to be able to tell us what the function might be. And don't forget that bioinformatics is such a new ground that is gaining every day. So, and this is a tool that integrates many disciplines into studying the genetic, into studying biological data. So you know that all over the world we have many data that is being deposited every minute. So ability to manage this and be able to analyze and utilize this for functional purpose is what we don't know. So, and don't forget that bioinformatics is now a kind of application of computer or computational programming, computation or application of computer programming in understanding, in utilization and analyzing of biological data. And one of those things that is going to gain relevance yours in, because I want to believe that yes, we have actually done bioinformatics, that is basic bioinformatics, where we came across BLAST, that is a basic local alignment sequencing tools. The use of that, I'm very sure that by now you know how to use cluster W to be able to understand how some proteins, how nucleotides can be aligned together in order to be able to make informed to be able to make an informed decision. And bioinformatics is such that is gaining relevance, application of drug design, application of the vaccine design as well is also gaining ground. And you know about genetic code. Don't forget that a different video is the fastest way of memorizing the genetic code, which you must know. So when you get home at your convenience, look for what AUG is standing for: CAU, GGC, CAG,